The MPhil in rare diseases has both taught elements and a lab project. In the first term, it was mainly lectures. So we had core lectures, which we went to with the students on the experimental medicine MPhil. So these were practical aspects of clinical research, epidemiology, and medical statistics. And then we had the genetics and rare disease modules. At the beginning, you cover more basic aspects of genetics. So, for example, genome structure, epigenetics, genetic variation. Then we covered, for example, the applications of genome-wide association studies and next-generation sequencing, which are becoming really important in the field of medical genetics, and I particularly liked this module. We also did several monogenic and polygenic rare diseases. Studying in Cambridge is definitely a really great opportunity because it's one of the world-leading universities, and the research carried out here is really significant, and I thought it would be a really exciting challenge to get involved in the research going on here. I chose the MPhil in rare diseases because firstly I really liked the structure of the course. I wanted something that had both taught elements and a lab project, and another reason I chose the course is because I'm really interested in the use of medical genetics in healthcare and in the clinical applications of genetic discovery. So I'm quite interested in the translational aspects of research. And the field of medical genetics is developing extremely fast, and it really has the potential to impact many aspects of healthcare. For example, increasing our understanding of disease and aiding drug development. And also one of the aims of medical genetics is to establish the concept of personalized medicine. And these are all topics I was really interested in, and so I wanted to learn more about these things on the course. And obviously I was also interested in learning more about rare diseases, which is one of the main focuses of the course. And actually on the course I realised that a lot can be learnt from the study of rare diseases, because often by studying a rare disease you get insight into more basic cellular processes which are related to also much more common diseases. For example, we studied rare monogenic forms of insulin resistance, and we saw how this has really allowed to get more understanding on mechanisms of of more prevalent forms of insulin resistance. Having the hospital near the university and having many important laboratories on the hospital site, that's definitely a very positive factor. I think it's very innovative that everything is going on in the same campus. No, so we were a really small group, which was really good because the lecture is very interactive. You could ask lots of questions, and sometimes they seemed a bit more like tutorials because you could really ask any questions you wanted, and I thought it was quite good that we were such a small group because it really gave you the chance to take as much as you could out of the teaching. I definitely think that the lab project that you do in the second term was the best part of the course. It was definitely challenging, but also really exciting and fun, and you just learn so much in such a short period of time, and it's over before you realise it. And I got a lot of support from my supervisor and his whole group, in particular from one of his PhD students. And we also ended up with some really exciting results, so that was also really good. I carried out my lab project in the Reed Lab at the Cambridge Institute for Medical Research. I chose my project because it involved learning how to use a new technique, which is known as BioID, and it allows to look at protein interactions in living cells. So I thought that was a really useful technique to learn and really interesting. And the aim of my project was to find novel interactors of two proteins involved in a group of rare genetic disorders known as hereditary spastic paraplegias, which are mainly characterized by axonal degeneration, which then leads to leg paralysis. And so we were looking at two proteins called spastin and DPY30. And we, first of all, did lots of cloning to create HeLa cells stably expressing two fusion proteins, which were these two proteins I mentioned, attached to each one attached to a biotin ligase, which you need to do in order to carry out the BioID. And then we did the BioID assay and then used mass spectrometry to identify novel protein interactions. And I think the project was definitely the best part of the course. It was quite challenging at times, but really fun and exciting, and you really have the chance to learn so much in such a short period of time, so many techniques and just translational skills, and yeah, and I got a lot of support from my supervisor and his group, in particular from one of his PhD students. And I think one of the best things about the project is that you really get a good idea of what it feels like to work in the lab, and to see the challenges that you have, that you get working in a lab, but also how rewarding it can be to get some really good results. So by the end of the project, we got some really exciting results, which was really good. We found known interactors of the proteins, which was obviously good because it meant that the technique worked. And we also found some really interesting candidate interactors, which I'm now writing about in my thesis. And yeah, and I think it's especially this structure of the course that has both the taught elements and the lab project is especially good for students that don't want to jump straight into a PhD after their undergraduate, because it really gives you a good feel of what it would be like to do a PhD and whether you like this kind of work and whether it's for you. I just really enjoyed the hands-on work and everything that comes with carrying out scientific work in the lab. So I've decided I'd like to do a PhD next year.